It's my pleasure to moderate this session on exposomic data science applications relevant to ADRD. And one of the first things I noticed about this was that the people speaking here have been using data science approaches for exposomics since before we were calling it data science or exposomics. So this is really something we have pioneers in these areas have been working on how to deal with all of this data. We've been talking about planning experiments most of the day. This group's going to talk about analyzing the extensive data that has been available. And so to start, we have Emma Cheminski from the University of Luxembourg. You may be familiar with her work with the Schymanski scale and going through and identifying things from high-resolution mass spectrometry and metabolomics. But she's really been a pioneer in cheminformatics, and she's going to talk to us about all that great work. So, Emma.
Thanks, Gary, and thanks to all of you for being here. I promise there is some caffeine in my slides in case you're getting sleepy. I know we're running out of time, but whatever you do, don't tell me to speak faster, because I will, and no one will understand a thing. My slides are actually available online, so if I do go too quick, you can download and check out some of the links that I'm going to mention. It's wonderful to be here, such an amazing lineup as well. And my blood is finally thawing out. So I think we've had this in several different ways already today. The lifetime problem that we have with exposomics, as we go through our lives, our profile is changing, but even our chemical profile or the chemicals that are available are changing as well. So this complexity in the exposome is extremely difficult. And just to kind of put it into perspective, in the 1960s, there were less than 200,000 chemicals and probably a lot less than this in the databases. Now we have over 200 million chemicals in the databases, and these are going up by about 20 million a year. So for anyone doing computational work on chemicals, this is an incredible expansion of chemical space. How do we find which ones are relevant? I guess this is what we're all trying to do in this session, what we're trying to talk about. So we all know we have this vast chemical space. I work generally with open science, so instead of going to CAS, which is under a license, I'm working a lot with PubChem. So this space is 123 million chemicals currently. I think we all know we have to look at this at different angles, right? So we have target analysis. This gives us very good reproducible results. We can tailor the methods exactly to the analysis, but these are, of course, limited by the reference standards we have. I've just named the mass bank examples here, 20,000 compounds. Whatever we can buy, it's not enough for this entire space. We have, at least in the environmental sciences, we have this suspect screening that gained in popularity. We've hosted the suspect list exchange since 2015, since I think before we were using exposomics. Widely, at least, the term was already around. By now, even in the Norman Suspect List Exchange, we've got over 134,000 chemicals, so suspect screening is no longer very small lists of a couple of hundred chemicals. And then we have the non-target screening, but the non-target screening in this space of 123 million is also extremely inefficient. So we've been doing a lot of work of trying to combine this down to a much smaller data set, PubChem Lite for exposomics. I'm going to explain this a little bit later. So of course, we want to expand the known space, the ones we can get the reference standard, have the fingerprints in the databases. Again, I'm just mentioning MassBank briefly here, but it's a proxy for all of the spectral libraries. And we are seeing a lot of data now coming in also from more the environmental side of the research to add to the metabolomics, so people are really contributing the spectra of the latest and greatest compounds that they're interested in as soon as they're getting the standards. And there's some actually very key contributors there that have relatively small contributions, but they're the only one that have that standard, and that is extremely valuable data. Then we have the suspect list. This morning I just added our two latest ones, and I was just before making sure that it was all going through the pipeline. So we now have 137 different lists on the suspect list exchange. We're now completely redesigning the website because our original concept didn't even work with this many suspect lists. And we're doing a lot of work now to break it also down into keywords so that people can navigate these subjects as well and compile the lists in different subject areas. And so it's still work in progress, but we've got a new website coming to help people do this. It's not just capturing the suspect list itself, a really important part of the work is also capturing the knowledge that's behind a lot of this, so the expert knowledge, and transferring this to other resources as well. So again, here we have the example of PubChem. We're also passing our information on to Comtox, and anyone who wants it, all the files are available. But we have a lot of lists with different subjects, so the EU cosmetics list is still one of our most popular lists by far. It's not just that they're cosmetics, but it's what the cosmetic was actually used for. We have things like collision cross-section, experimental data coming in, so there's also a central collection here so that people can download this and improve their methods. And I'm going to mention this a little bit later, a lot of transformation product data as well, because I think it's already been mentioned today. We have a lot of data, but of course, when all of these compounds hit either our bodies or the environment, a lot of them will metabolize, they will transform, and we need to also be able to capture these transformation products as well. We have been working on a new website, so we call this one FairTP. So this one is the website of the known transformation product data, so people can really just focus on what data is available for specific compounds, so the numbers look a lot smaller. It's very interactive. So again, we've got 11 different data sources, most of them the norm and suspect list exchange, and you can see for every compound actually how much data we have and where we have it, also the metadata associated with the reactions. And we're doing a lot of work on making not only the images available so people can use it in their presentations, but also making sure there are download files that are compatible with people's workflows so they can actually download these lists and screen them in their samples as well, especially if you want this really specific data for certain chemicals. So of course, our known unknown is this huge challenge. So 123 million records to go through is extremely difficult. When we first released MetFrag, it was designed on PubChem that was 25 million, so now we've got 100 million more to go through. It's incredible how quickly these numbers are going. I'm not even going to talk about the PFAS today, we can do that in the coffee break. What we generally see though is we have very few candidates with high information content, these are usually the ones we want, and we have this very long tail of candidates with low information content. But the one we're interested in might be in that tail, and this is our problem. We still have to kind of somehow screen through them all. So we're looking at different ways of prioritizing how can we get the interesting bits out of the tail without having to go through 3,000 candidates if all we want to do is identify nicotine in our sample, because we know nicotine is in most of our dust samples by now. We don't need to go through 3,000 candidates. So with PubChem Lite for Exposomics, we kind of took a look at the table of contents in PubChem and broke it down into the sections that we really want most interested in. So we have a lot of subjects on the environmental side. We have the Exposomics category. This is also where CTD is in. You're going to hear about this in the next talk. We have the suspect list kind of hidden under this category. We're thinking of a different way to get that in right now. And then we also have the metabolomics data set, so pretty much the key data for our subject area of interest. The best thing about this is if they're in these categories, it also means that we have information on those records why they're in those categories. So not only do we have a hit, we actually have some kind of information to follow up on, which is also really important at Exposomics, that you're not just following up a lead that will absolutely go nowhere, and you're spending a lot of time. So this is distilled this down to 500,000 compounds. We're trying to trim this down a little bit at the moment. There's some noise coming in. And we've actually released a website for this to help people navigate it, because it was just a very fair in the data sharing way, a CSV file on Zenodo, but no one could actually browse the data. So Valentine has done a really nice website for this. We have just the very key information for the mass spec, so we have the mass and the X log P for the retention time. But we're also demonstrating the annotation content as well. And this is where the context comes in. We have the chemical stripes. I'm going to come back to this. Just watching that, I'm talking not quickly enough by the looks of it. And we also have the annotation content, so you can actually see how much information is available. And these actually really help very quickly see whether this chemical is something of interest, or if it's something that was maybe of interest 30 years ago but is not of interest now, or it's just one of these data blips that is just coming through, maybe not enough data, and then you can look and see if this is one of those up-and-coming chemicals or just kind of one of those noisy things coming through ECHA. We've also started building some different searches so that people can browse this. So if you're interested in certain substructures, you can now do a substructure search. You can download. Our download files are actually sorted by the annotation content, so you're kind of getting the most interesting compounds towards the top. Capped so that you don't blow our servers. If you want more, let us know. The data files are always there. And we've also got collision cross-section search, which is really nice at narrowing things down to very small files. So getting back to the Parkinson's context, how do we actually do this in the context of Alzheimer's, dementia, any form of neurodegenerative diseases? And this is an example from reduced penetrance of the LRRK2 mutation of Parkinson's disease. What chemicals could possibly send someone with the LRRK2 gene into the track that will develop the disease versus not developing the disease? And considering all this in the timeline of our chemical space expansion. So it's a very small data set. Our workflows are very complicated. I'm not going to go into it. We're doing all the screening. In the end, we were getting 1,000 chemicals coming out. How do we then choose out of these 1,000 chemicals which are the ones we want to look at further? I put this slide back in because Gary said something on Monday when he was in Luxembourg that he's really worried if people are doing suspect screening, you're kind of in a lens and you're missing some of your data. And when we actually went back through those 1,000 chemicals and looked at them and looked at the significant one with the lens of different suspect list, we saw exactly this. If you're just focusing on one, you're actually missing some of the significant chemicals, and they're actually going through pretty much all of our major categories on the suspect list exchange. And if we went through the categories that we have in PubChem Lite as well, same thing. We're really seeing significant or potentially significant chemicals hitting up against all of these categories. The one that we actually filtered out, again, my poor student had to publish something in the end and we've got 1,000 chemicals. How do you choose? When we were kind of looking through that, dropping it down in a lot of different ways, we had 2 filter out that really caught our attention because these are hazardous chemical replacements. And we're thinking in the context of dementias that develop over 10, 20, 40 years, these are kind of worrying already. So one of them was actually, okay, the animation is messed up a bit, I'm sorry. So one of them was bisphenol S, which we've heard the bisphenols mentioned today, and another one was PFBS, which is a substitute for the PFOS and the related chemicals. And if we actually look at the data, so I know it was mentioned this morning already, the BPA should have been restricted for a long time, depending on which lists you look up and when the restrictions, the dates are a bit contentious. But if you look in the patent data, we can actually see bisphenol A has only been a big hit in the patents in the last 20 years. Bisphenol S, actually a much shorter time frame. So this kind of triggered us to look a bit more into the patent data and can the patent data actually reveal some of the behavioral changes that are going on? Because we'd seen a lot of these stripes and a lot of them kind of had this pattern. We're building up to a lot of data in 2022. Then we get this cutoff at the end because it takes a little bit of time for the patent data to come through. If we looked at PCBs, however, and PCBs were mentioned this morning, these have been regulated for a long time, you can see a very, very different pattern in the patents. You can see we never got this increase in patents in the last couple of decades. Then when we looked at the PFOS, or the initial PFOA list that was on the Stockholm Convention, we also started to see this tailing off as well. So this was regulated only in 2009, if I've got the date wrong, if I've got the date right, sorry. You could see it tailed off, but they actually had to release an updated list a few years ago because they were noticing people were kind of evading the regulation. And you can see for the substances that were then put on the list, the patent numbers were going up. So this is really telling us, I think it was the speaker this morning, regulation is working. We're also seeing this in the patent data. So we contacted our computer science department, can we do some network analysis to find the next problematic chemicals? And this was actually super fascinating work. I'm apologising for the change in colouring, but I couldn't do terribly much about it. One of the chemicals that popped up was actually one transformation product away from a chemical that Beggar also found in the dust related to the penetrance, where we saw an underexposure in the LRRK2 mutation holders that did not develop PD, and we saw a marked increase in the ones that did develop PD. And the other interesting one that filtered right up to the top, this was driven by a PFAS list because this analysis is quite intense to do, the patent set is huge. The one that came up at the top was actually not a PFAS at all, there is no carbon in this molecule. But the interesting thing was already at our CTAC conference in 2023, Daniel Sahn, who is a guy working with Torsten Riemsma, really driving the analytical methods for very polar compounds, he was already noticing these compounds coming through in his samples, coming through wastewater treatment and saying these ones are not responding to drinking water treatment. And he was worried about them because if they're not responding for treatment, we don't want them increasing in the environment because we don't know what to do about them. And when we looked at the patent data, this was going through the roof. We were having data, so we had this, always had this lag. We had data streaming in from China for these chemicals. We were like, okay, what's going on here? Doesn't take you very long to see that they're associated with electric vehicle production. It all started to make sense, and we've got our next PFAS substitute coming through. This is why we're at conferences, because Selena Tisler, who's also doing methods right on the edge of the analytical boundary, so she's working with supercritical fluid chromatography, she was listening in, developed methods, started a collaborative trial across Europe. We're finding all of these compounds throughout wastewaters in Europe, and she actually found this really nice arsenic fluorine species that Daniel hadn't had on his radar at that stage. So this is why we're sharing science at conferences, sharing the information. So one of the challenges I want to ask us for the discussion panel a little bit later is also how can we use the data science to see how much of the exposomic space can we actually measure. So if we actually go through some of these timelines, you'll see a lot of these discoveries have coincided with the analytical ability to measure them. And there's some of these chemicals that are right on the edge of what we can measure. There's probably a whole lot of them that are beyond the edge of what we can measure. How can we actually use this open data and the sharing and the knowledge in the community to drive the analytical innovation to help us answer our questions? So with that, I actually did manage to stay on time. This is amazing. Thank you very much for listening. Thank you to all of my collaborators. Looking forward to the discussion.
And we're going to hold questions for these 3 short presentations to the discussion panel. So next up, we have Carolyn Mattingly from the NC State, North Carolina State University. She's going to talk about the comparative toxicogenomics database. And often in data science and AI, we hear about things about garbage in, garbage out. I can't think of someone who's done a better job of preventing garbage from going in. And I think it has to do with the curation of that data coming in, and she's going to tell us some about this work and the fantastic analyses. So, Carolyn, take it away.
Great. Well, I guess it's good that we're not garbage. No, that's fine. Yeah. And I've been self-diagnosing today. I'm in severe cognitive decline with some pretty bad jet lag. So if I start garbling my words, I apologize. So I'm going to talk to you about the Comparative Toxicogenomics Database, or CTD, because it's kind of a mouthful. We've been building this database for longer than I care to remember. But basically, the goal of this database is to try to integrate information from the literature in such a way that we can help facilitate understanding about the influences of environmental chemicals on disease and what those underlying mechanisms might be. So we do this through manual curation, which is it's not being replaced by anything, but we're actually integrating a lot of automated methods because, as you can see from the scope of the data and the landscape of data out there, it's very hard to keep up to pace with certainly the literature, but also a lot of the analytical data that's coming. So I'm just going to spend one slide explaining the curation, what this looks like. So again, we take this information from the published literature. We do not evaluate the quality of the data, with some exceptions. And this is becoming a lot more challenging, I think, as we're seeing AI insert itself in paper writing and such. So essentially, we have 5 curated types of relationships, and this is all kind of distilled. There's a lot that goes on under the hood here, but we're interested in chemical disease relationships, chemical genes, and when I say genes, I'm also talking about proteins, epigenetic modifications, anything that is sort of involved at the molecular level. Gene disease relationships, chemical phenotypes, and what we call phenotypes are essentially biological responses that would precede or coexist with an actual clinical diagnosis. And then we have exposure data, which is sort of like those first 4 are really what we call our core mechanistic data. We also have exposure data, which is sort of the more recent addition to the database, even though it's been more than 10 years. And that involves very deep curation of a lot of epidemiological studies, what exactly was measured, what levels of measurement were detected in those studies. And so how do we do this? We have mechanistic or automated ways to triage the literature according to criteria that we have. We have some help with PubTater group at NCBI, which has been really helpful for doing entity recognition, helping our curators identify essentially the big picture concepts, and it greatly increases the efficiency of their work. And we're collaborating also with some computer scientists at NC State who are experts in large language models, and we'll have a paper coming out soon, I hope, that will talk about essentially where they're falling short on trying to approximate the data that we curate manually. And there's some good news, but there's also a lot of bad news there in terms of we're not going to be able to rely on, if we're trying to capture the complexity of exposure data, the tools are not quite there yet, in our hands anyway. So we've been around a while. We've had have a lot of increasing data. I'm not going to read you the stats there. As far as neurological disorders, so we curate all of our information with standardized vocabulary, so everything is controlled, used in a lot of other public resources, which allows us to integrate with other resources that are out there. They're also hierarchical, and so this can be really helpful when you're looking at chemical categories versus specific chemicals, as well as with diseases. So you can find information on Alzheimer's disease, but also higher level dementia categories. So we have quite a few points, about 3,000 different neurological disorders that have curated data in them, and we can show interactions or overlap with over 6,000 chemicals. And of course, based on Emma's data, you can see this is a small fraction of the chemical space, but again, we're dealing with different sources, so these are our published data sources. So we have basically our curated data, which are in some cases binary relationships, in some cases quite a bit more complex than that. But it's really the power of this, and I like the intentional science comment earlier this morning that, individually these papers may find one connection, which it's sometimes hard to understand the broader context of why that would be important, but when you're able to integrate all of those data together and start asking questions across different types of entities, you can start to see different patterns. And so these are just examples of a simplified version of how we can integrate these data and develop inferences about connections between more complex information that you would rarely find all of those details in a single paper, for example. So the inference networks, we call them, you have a chemical that interacts with some gene. If that gene has some association with a disease, we can sometimes make those connections then between a chemical and a disease. And depending on the source of the data, you might be looking at an epidemiological study, and they might see a connection between a chemical and a disease, but you may not have that intervening mechanistic information. So by having epidemiological data, as well as a lot of the experimental, more mechanistic data, and pulling those together, we can try to help facilitate some hypothesis development. Then we also have what we call tetramers, and this takes that level one step farther, where we integrate phenotypes as well. And so if you're familiar with adverse outcome pathways, this is attempting to understand some molecular initiating event that may be affected by a chemical, and there are ensuing phenotypes that could lead to a disease. And so we essentially have all those components, and so helping users put those pieces together and get a better idea about how those data all connect. So data is accessed through a public database. It's freely available. And typically people are coming to the database with a disease or a chemical in mind, but we do have a lot of analytical tools and more advanced ways of querying the data. And you can always contact us if there's information that you need that you can't really figure out how to get to. So I'm just going to show you a few views of the types of data we have and what this might look like if you're thinking about neurological disorders. So for our population-based exposure data, we have, we're at the mercy of what has been published in the literature. So sometimes you need to go to more generalized categories of chemicals. So for neurocognitive disorders, which would include Alzheimer's, you would learn about the demographics of the population, what levels of chemicals were measured, what outcomes may have been observed, whether those were statistically significant connections. And of course, these studies are coming from across the world, so you can sometimes see interesting differences between where things have been detected, for instance, and where you might see disease incidence geographically. So the experimental data, this is our Alzheimer's data, so chemicals versus genes. So your chemicals are on your left side, genes on the bottom, and you can see kind of where the data are clustering. And again, these are not dissimilar categories as we've seen earlier that have been associated with Alzheimer's. And then, of course, the genes that tend to cluster with these different environmental exposures, we have tools for saying, all right, this cluster of genes seems to be showing up with Alzheimer's, and what do we know about how those genes actually interact with one another? So for chemical disease information, we'll have curated relationships, but again, a lot of times, depending on the source of the data, you might not understand necessarily what that connection is based on. It's a little hard to read, but on the left column, you have chemicals, then you have diseases. If there's some context in the column that has the red arrow, that says there's actually curated data for that. But we also have these inference networks, which I mentioned earlier. So in this case, this is cadmium related to Alzheimer's disease. And we would make that association independent of whether that's in the literature or not, based on the fact that there are 80 genes that have been associated with both cadmium and with Alzheimer's disease. And then we have integrated a number of tools that can help you sort of think about why would those 80 genes be associated with both cadmium and Alzheimer's in this case, things like enrichment analysis, and also some molecular pathway function if you're wanting to understand, are there known interactions between these genes? And it's not just that they've been sort of thrown together. Some of the data in CTD I think could be really useful for thinking about comorbidities, and we've heard about diabetes and cardiovascular diseases being associated with Alzheimer's. And we have tools which we call comparables, so you can look up any disease and say, based on the data that are in CTD, what are other diseases that seem to cluster with those groups of chemicals or those genes? And then this is sort of how tetramers look. So this was taking these inferences and then looking at chemical, gene, phenotype, and disease relationships. And so there's a lot of evidence that inflammatory disease or inflammatory phenotypes are associated with Alzheimer's. So you can query very complex relationships. This is a pretty stringent query, so sometimes you have to iterate on how you're querying the data, but this is basically then taking all the data in CTD and saying for Alzheimer's disease, if you're interested in inflammatory phenotypes, at the top in the purple is the grouping of phenotypes that are associated with Alzheimer's, all the genes that are associated with them, and then chemicals that interact with those phenotypes and genes. And then just another view of this, which you can't generate this in the database yet, but you will be. And these are clustering essentially on the left. You're seeing chemicals, then you're seeing genes in the middle, and then clustering those genes into different phenotypes. And this is telling you things like oxidative stress, mitochondrial dysfunction, things that we know to be associated with Alzheimer's disease, but we don't necessarily understand all the genes that may be at play there. So we have lots of challenges to us, not the least of which is the data space. So I think AI is going to start to play a big role in thinking about how we can have a greater return on investment in all of the publications that are funded through all these mechanisms. And by integrating them all together, I think we can sort of propagate their impact in different ways. There's lots of other sources of data that we do not yet include, so things like in the States, the EPA, the FDA reports, things like that. A lot of them are not in formats that are so accessible, and so dealing with a lot of the FAIR standards. So integrating across other resources and making sure that we're all using data standards that are going to allow us to connect better to each other. And yeah, just thinking about the complexity of the data. And I think these kinds of meetings and other meetings that have been going on, really having more of a community where we can bring our data together and build tools on them so that we're not reinventing the wheel each time we sort of evolve this field. So I will stop there to keep us on track. And just to say we have a lean but mean team working on this project, and we haven't exactly had more than a 5-year grant period, but we've had many 5-year grant periods put together, and I fully support the idea of very long grant periods.
No detection of jet lag there. Excellent job. So next up, we have Dinesh Barupal from Mount Sinai. Dinesh has been working to pull that data off those mass spectrometers for many years. He's created many great databases and tools like the Blood Exposome database, and he's going to talk to us about one of his newer tools and databases available. So take it away.
All right. So last talk, I started the symposium with the workshop. Today morning, we did on the untargeted exposomics. So last talk, I'll talk about exposome correlation and interpretation database. So we want to gain mechanistic insights from Exposomics core human biomonitoring data sets. So we have a lot of discussion about what exposome is. This is my thought on the chemical view of the exposome. We take IARC monograph as a representative catalog of exposures. There are over 1,100 agents in IARC monograph. 80% of them are individual chemicals, which means that they are connected with chemical abstract surveys or chemical structures. Rest are complex exposures, but they're still chemical complex exposure. So overall, we can say that 90% of the exposome is actually chemical exposome. From where these chemicals coming? Majorly there are 8 categories. So we have anthropogenic sources, natural substances, drugs, consumer products, food, metabolism, and human microbiome. And that pie all together can have hundreds or millions of compounds there. But what's the relevant is the blood exposome, that what gets into the blood out of those, that's the relevant part. And we need to focus on that one because that is the equivalent to the genome. We can analyze blood, we can analyze genome. That's the equivalent entity, blood exposome. So I want you to remember exposome, chemical exposome, and blood exposome. That's what we want to focus. So common question in the chemical exposome field, which chemical exposures, what are their levels, how are they processed in the human body, what are they doing in the human body, how they are related to each other and other molecules, how are they related to human diseases. And so all the answer to these questions are now we are at the level of exposomics knowledge bases, where we're systematically curating and organizing answers to these questions. So CTD is an example, we have other databases. So I'll give you a diagram that what's the landscape of exposomics data repositories and knowledge bases in the US. In the US, we have this program called DRKB, Data Repository and Knowledge Bases. It's managed by the Office of Data Science and Strategies. And data repositories are databases where people submit primary data. So we have genomics data, proteomics data, biomonitoring data. And we have specialized repositories and generalized repositories. From there, we get core exposomics data sets, and we also get a lot of primary literature when people analyze them. That goes to knowledge bases. And we have an exposure knowledge basis, and we have toxicity knowledge basis. So on exposure knowledge basis, we have example like geospace, CTD also exposure knowledge bases include exposures, blood exposome, and now we have a ECID exposome correlation and interpretation data. I'll talk about that. So remember, we have data repositories where the primary data goes, and we have knowledge bases that what the knowledge from these data repository core data sets, that's what we're curating the knowledge base. And it's part of DRKB network that manages, coordinate all other resources like UniProt, gene ontology. It's about 50, 60 resources across the whole NIH data science system. So what's the idea behind ECID? So ECID is a biomedical knowledge base of curated interchemical correlation. It's a very simple idea. So what's the correlation between two chemicals inside the body? From core chemical biomonitoring dataset in Exposomics, selected from only human biomonitoring studies, no animal studies, only human studies using targeted and untargeted assays. So in simplest way, think about this chemical A and B measured across multiple samples. We're computing a simple experiment of Pearson correlation. If that correlation is high enough, we want to interpret in three ways. That is this really a co-exposure? Or is it a metabolic transformation, or it is indicative of a toxic effect of that exposure if it correlates with the metabolite. So three ways we're trying to explain the interchemical correlation. So what is a biomonitoring data set? So, Emma really highlighted nicely that what is a targeted approach and untargeted approach, but always simplest we can think about that when we have a targeted, we have an analyzer here that actually screen that what exactly targets we want to record for a blood sample. And these are panels, like panels of PFAS, panels of phthalates, using a good example will be CDC enhanced data sets where over 150,000 samples have been analyzed by different panels. On the other hand, we have untargeted approach where analyzer actually doesn't filter the data. All the arrows or signals from a blood sample are all collected together into a data file. We can have up to 10,000 signals for one single sample, and but we are doing that by high-resolution mass spectrometry that allow us to do untargeted assays. So architecture of ECID is simple that we have repositories that provide us the core data sets. We select the core data sets. It goes to CCDB module, chemical correlation database. We have a Exposome data interpretation resource that we compile using knowledge and ontology available in other databases and primary literature. And then we do the curation and select those correlations and add into our database. All the data are publicly available on Genedo, on our websites and GitHub and the lab websites. So just a quick statistics about it, that how many data sets we have right now. 112 total data sets. Again, these are human biomonitoring data sets. And we have targeted data sets, 63, 10 CDC enhanced, CHEER here 24, and ECHO program 29, close to 150,000 samples covered by targeted assays. And untargeted side, we have one CHEER here study, ECHO program 23, and a lot of studies from Metabolon. These are the inclusion criteria that we should have at least 50 human sample, blood or urine. Spectral data, MSMS, must be collected. It has to be US-based study. That's one of the exclusion criteria we have right now. Inclusion criteria, sorry. And at least one of the high priority substance chemical is included in or annotated in that study. Because we have so many studies where no annotations, we can't use them. So I have to have some exposure chemical annotated in a study. So correlation statistics, we have close to 4,000 reported chemical in targeted and untargeted assays. They cover exposome, lipidome, metabolome altogether. 665 out of them are high priority chemical exposure. That we went through them and we said, yeah, this is a good exposure. It shows some sort of exposure among those 4,000. 135 reported only in untargeted data sets. They were not covered by targeted. And we have 9,140 unique correlation from 311 chemicals that were selected, and we pass it through our interpretation pipeline. How we define our high priority is that a chemical needs to have some evidence, published, in silico, observed, of toxicity, so we can then put in the priority that this is what we're going to follow up. So we have a nice website for exploring the data where we have a chemical list, we provide summaries and detail about basic information and direct link to the curated correlations. And you can query by INSERM key or name, and it will give you the first column that how many data sets have that same correlation observed. So like if you query PFOS, it correlates with other PFOS or other chemical and give you that how many data sets have the same correlation. And then we have the interpretation, AI generated interpretation, but we curate and expert verified. So AI generated, expert verified interpretation. That's the correct interpretation of the correlation. We rely on AI heavily to process all this literature data quickly and getting all these summaries. So we rely on Gemini 3 Pro Google model, and we have our own PubMed full text index. And whenever we see two correlations between chemicals, we pull all the publications with PMIDs, and we get a really nice non-hallucinated summary of the correlation. We force the model that do not create answer if it's in the published literature only then. And so we get all the PMID cited inside the summary. And if there is no literature, then we ask the model that what's your suggestion about it? If there is no publications, then we still ask the model that give a summary that might help a new user that, okay, I see correlation, nothing reported, but what could be the possible way we can explain the correlations? So this is one example, PFAS in CCTV. So 3 common PFAS, PFOA, PFOS, and PFHX. And what are the chemicals correlating with these 3 PFAS? And it's a metabolome data sets, and we see a lot of these chemicals correlating. Some of them are exposure, like this is a pesticide-related metabolite, and these are many endogenous compounds that correlate with PFOA. So what we did was we took the spectral data from metabolome, we reprocessed everything, because this is untargeted high-resolution data. There's a lot of things that they have that they did not annotate. We reprocess, and these are additional things we are seeing by using the software we develop. And so we have our own internal pipeline that takes the raw data, reprocess everything from scratch. And so by doing that, we are getting additional PFAS that they miss, not annotated. We're getting additional pesticide metabolites. We're getting additional endogenous metabolite signatures that correlate with PFAS. So that's how we are expanding the correlation database by looking at the old data, reprocessing it, capturing all the exposures. So that's how we are developing CCTV. 3 minutes, right? Yes. So it's all backed by this called Exposome Data Interpretation Resource. And so by developing this, we have a list of about 60 databases that we utilize. These are government databases, committee reports, a toxicological database that we pull together. And whenever we try to interpret or prioritize a chemical, it goes through these databases. And while we have access to all that, we use this to develop new softwares that helps in toxicity and annotation. So this is one example, in silico predictions. So we use quantum chemistry approach, we use machine learning approach, and we are now using Google Gemini directly to predict spectra. And it's really like Gemini actually generate very high quality spectra compared to quantum chemistry or other machine learning methods. So we have a paper under review right now where we show the example that we can simulate 10,000 spectra, really high quality spectra in one day, and it costs about 10 cents per spectra to generate that. And we will be able to solve this problem of that many chemicals don't have spectra. That problem will be solved very soon because of these tools are getting really accurate. We also work on the in silico predictions because many correlations reflect reactions. And we have a correlation, M over Z value, a peak, but what the reaction might be behind it. So we end up generating I would brag about it. This is the best way to generate in silico transformation product. We tested it. It's covered 100,000 reactions available in PubChem. And it uses machine learning, template-based approach, very chemistry-oriented, explainable way that reaction happens. And we simulate transformation product for any exposome chemical, and we can look back in the correlation data. We also have models for in silico prediction of toxicology. We published this paper using Tox20 programs data and other bioassays data that if you annotate a compound and there is no paper out there, where do you go? And so we need to do this in silico prediction of toxicity and to get some idea about that what a chemical might be doing. 1 minute. So yeah, my lab develops software, a lot of software for exposomics, and we have a lot of databases maintained, and these are all freely available. We support open science, so you can go to website idsl.me, and that's where we list all the software and databases. And if you want to learn really more, you have to pay $2,000 per week. And we do a full intense course. And the next will be in 1 month, but we also do in February course. And with that, I'd like to thank my team, my collaborator, and NIH for funding for last. I don't have multiple 5-year grants, but yeah, we are in the phase 1 right now. And thanks. Right in time, right?
Yeah, so we are going to stay on time because I literally don't want people to miss the boat. So we're going to end at 5:15. So we can both take questions from the audience if there are any questions from the talks, and we'll get into some discussion. But I see a hand up already, so why don't we go to the audience?
Thanks, Dinesh. This question is for you. Interesting talk in terms of using large language models, but I'm curious in the sense of your choice of Gemini. Why not other basically large language models? So you're using a proprietary-based model, so it's not open weights. So fine-tuning and training might be a potential challenge in terms of using the proprietary models. But I'm curious in the sense of why the choice was made that way. And my second question is just how do you see patient data sort of supplement the work that you're doing?
Okay. So we went by the model accuracy, performance. So we did benchmark over a local model, publicly available machine learning model, and Gemini simply outperformed it. And we thoroughly tested it. And that's why we thought like, okay, well, if Gemini works, we don't have to train this local model anymore. So the problem we have, the spectral prediction, which is a less sensitive problem in terms of like data ethics and data sharing that, yeah, we can just ask them, Google that predict spectra for the smile. It's not like a sensitive question. And it just gives the spectra back. So that was our decision that it just performed well compared to other models. What was your second question, Nabil?
Regarding patient data, I would love to see more clinical sort of picture emerging.
Yes.
And that will help us as clinicians.
So at Mount Sinai, we have our Mount Biomi cohort, a million Mount Sinai cohort, where we do have the biobank sample and longitudinal electronic health records, and it's happening. All those samples being analyzed using high-resolution mass spectrometry. So we can do all that analysis where it's a EHR-connected biobank, and we can look at the exposomics on those biobanks.
So, Dinesh, a few days ago, there was a paper in Nature Metabolism, and the title was Why Machine Learning Fails at Mass Spectrometry for Small Molecules. Thoughts? That was like 5 days ago that came out.
So it's really the lack of training data. So when we train the mass spectrometry train a machine learning model, there is just not enough training data. And so in Emma's talk, you say that about 25,000 compounds have a spectra. That's not enough. That's like very tiny fraction. If you train model using a very small training data that doesn't cover the entire chemical space, the accuracy will be lower. But it's too costly to have like $1 million or 1 million compound spectral library. That was the main reason why the existing machine learning model fails. While large language model, they rely on these inbuilt rules. That's what they're learning, that how mass spectra are generated and fragmented. So they don't rely on training, they rely on rules. So once they learn a good rule, it can be applied on any chemical. That's why they are successful.
So you think like the idea of using large quantitative models for spectra should totally work?
Yeah, I think so.
I agree. I think there's a lot of potential there. I mean, I think the combination of the LLMs and looking at the literature is a separate thing than the LQM sort of approaches where you're looking at the data, where you have all this massive data coming in. But it is like the type of training and what you're training for has to be fit for purpose in this situation.
Exactly. If we go back before all this computer, in 80s, people were like trying to simulate a spectrum on a paper. I look at the spectrum and I design how the fragmentation will happen, and they will say, you'll see this fragment, that fragment, that. Now LLM is doing exactly the same thing. So LLM become that level of expert that it can actually walk through a structure and create the fragmentation pathway, and it's amazing. It's really to that accuracy.
So Emma, what are you thinking about like with these data sets and the training of these potential models? Like obviously your PubChem Lite was trying to distill it down to being more manageable. Do you think there needs to be more distillation, or are these systems powered enough now that they can handle these larger numbers? I mean, like 3 or 4 years ago, it was very different.
So it depends if we want to talk about the carbon footprint of AI, which is actually an interesting discussion in a lot of environmental conferences. If we skip the water use and the electricity use, I think there's a lot of interest in the results coming. So I actually had a talk with Dinesh over lunchtime and emailed my team asking them what they thought about some of what he'd said, and they have questions for him immediately. Because we've had a lot of requests for PubChem Lite put out the spectra, and a lot of the older methods have been around for a while. You can see these newer methods coming, but they're moving so fast. If we would do it now, in 2 weeks' time, it'll be out of date. How do we keep that up to date? So like the movement in the field is so incredibly fast right now. And it's interesting what he said, because from the machine learning point of view, like even if we would have Metlin, so we do have the million spectra somewhere, or at least someone has, that he's not sharing. If you talk to Juho Ruusu, who was one of the guys behind the CSI finger ID, he doesn't think that would even help solve, because it's still such a small fraction of the space we have. So it's really interesting. I would be interested to see his data set, because this is our worry also with the tox predictions. We have such a biased subset of PubChem. How do we know that that will extend to the entirety of PubChem? That's why I kind of pointed the question. It's like, we know what we can measure, but how much of that can't we actually measure? And this is going to behave completely differently because we haven't seen it yet.
It is interesting thinking about the carbon footprint of AI use, which is something I think should be discussed, is that I don't think it's much different than it was if you go back 20 years ago and you were doing experiments, is that you try to think and plan experiments well to maximize the use of the resources. And I think a lot of people are using these LLMs and different systems like to help get ideas for what to have for dinner tomorrow night or something. Like, are these really uses you need them for? And I'm sure there'll be a bit more gatekeeping as, the users are there and there's some more subscription models. I think that's coming. But even now, it seems like a lot of the things that we're doing, I think this is the first space where we're really getting these areas that are really pushing the limits from a computational side compared to what they're doing, like in high particle physics and other things. But it is something that it is, I think you should be very thoughtful in the design of those experiments and not just use it because it's right now free on your desktop.
It was actually an interesting first question we had, because it's a really challenging time to do open science now for exactly that reason. The models that are moving quickly are no longer open. And then we're really stuck. And then you've got people like you and me who've been developing open data, putting it out there for everyone, and this is now being milked and everyone can have it, and it's really hard to get the resources to do what we're doing.
Right. And I think
So
this is
this is the other downside, is like, how do you incentivize actually all of those people generating the data to get this into a way that the machines can talk, and then everyone profits from it, and it's in the end our students who are like manually doing all this metadata curation.
Even
So
like
there
on
is a
the
downside.
copywriting like of literature, people have been saying they've been using it to the LLMs mine copyrighted material because it's in the open, even though it's copyrighted. And while there's been lawsuits and things, I don't think that's resolved. So, Carolyn, what are you thinking about? Again, like you've created these open source things, and now people can just go through and mine it. Yeah. Which, if they're academics doing it, you're like, yeah, this is great, but is it others? I think they turn it on automatically.
It's on, okay. Yeah, if you go on ChatGPT and ask questions that I know we have the answer to in CTD, and I say, use CTD data, it's very hard to see what the source of the data is. And so I've tested it, to see if questions that I know we can answer, where are they pulling the data from? And you can't actually get reliable answers about that. But so it is being incorporated into lots of other sources. But to the point about the standardization, I think trying to get that, so I should qualify, we're using LLMs for summaries too, and if you can give the systems very standardized, structured, reliable data, they do a great job at that. If we try to get the LLMs to curate the type of data we're curating, it falls short, but a lot of it, I think, could be solved by really standardizing the data that are in the data sources out there, that are in the publications. And, we can't get publishers to require standardization of terms like genes and chemicals and things like that. And so without that standardization, there's just a lot of poor information that comes out of some of these automated approaches.
So this gets into like some of the needs for the field on harmonization, standardization. Mm-hmm. What do each of you think are some of the most important things that are needed from a field perspective? Focusing on exposomics, but it can go broader out to biomedical research.
Yeah, I think there's some pretty low-hanging fruit with, the vocabularies are out there. And of course, for a lot of the emerging chemicals, those are going to have to be incorporated in standardized ways. And that may be a little bit more challenging. But for information that's out there now, at least in the published literature, there are standardized structures for chemicals, genes, diseases. People don't use them, though, and in a lot of cases, they don't know about them. And so I think some of it is educating the scientific community, but I think, going back to microarrays, standards were required, and people followed them, and that made a huge difference. And I think we have to do some similar types of things with what the products are that people are generating and making sure that they're consistent with each other. And I think we could get a lot more out of the collective
I think this is something, too, when you think about who drives that. Like with a lot of the DNA work, the publishers got on board to say you had to report these things. And we'll often look to our funders to say, can't you just put it in the program announcement to say these things must be done? But that's not a simple thing to do, and I appreciate that. But it's really in thinking about if these things are so critical, who are those key gatekeepers of this, and how do we get the community to take advantage of them? I think from a publishing standpoint, like from a journal, like they don't want to put a bar up too high because then the submissions go down, and just the whole system there is so volume profit driven at this point. Like I think there's going to be so much resistance to that, which this could be a whole other topic about that. But so other thinking about harmonization or other areas that you think are needed.
I was going to say, if we just take the example of GC versus LC, right? NIST or the EI GCMS data was standardized very early on. This NIST was available. If you want to annotate in GC, everyone just buys the database. It's a reasonable cost, and it works. And LC hasn't been standardized. We have 5 vendors in the works, and they all kind of want to do their thing. And there was some point when we were kind of hoping they would kind of align, and now it seems that they're actually actively making it difficult to even get good MZML files with ProteoWizard anymore, which will make all of the open workflows break. So it's like we're kind of seeing a swing back against a lot of these efforts because they want to drive people to their software and make more profit because these are harder times. So I think this point that there's also this profit versus the good of science. And how can we fly above this? And I think actually the funders would have a big role, and we've just seen our funder roll back some of their regulations to make it easier for people, but then we as scientists no longer have the argument to say, we have to do this, our funder requires this. But on the other hand, now you can see what's happening with the profit taking when we're required to do things and some people are taking advantage of it. It's not easy.
Yeah. I wanted to comment on that one. That the data harmonization problem, it needs to be handled on data repository level, because nowadays we submit data to a repository, it should be handled at that level. And a repository, a national repository of each country or region can do it because we know how to do it. It's mostly the implementation of how to implement it. So especially the untargeted exposomics data, they can be harmonized, but it needs to be done on the repository level. Data repository level.
And so are current repositories up to the task?
Yes, but they're not doing it. Yes, yeah, they can do it, but they're not doing it.
Can I just make a comment? So as part of the MAHA agenda at the NIH, which I'm providing leadership for, there's a big discussion about how we bring different data, harmonize different databases, because right now it's a real challenge. So there are groups of scientists across the NIH and the biomedical spectrum that are talking about data standards and how we really need to be reinforcing this. But astonishingly, and I can't understand this, but there are AI experts that are coming in and basically saying, by overlaying standards on the way that we collect and manage data, that leads to a compromise in innovation. I don't know if there's anyone in the audience that actually can speak to that or wants to introduce that topic area to this. So there are people who claim, bring AI on, and AI should be actually this powerful tool to bring data harmonization across. So I don't know. So
I think on the standardization side, I think we've seen, like, if you standardize all mass spectrometry across the world, you would drop innovation down. But that doesn't mean you can't harmonize and have common reporting. And so I agree with that. With the AI models, you want to give them all the data, but it's kind of like the combination of targeted and non-targeted, is that if you do both of them, you can anchor it in some reality, but then let the AI go with the blank slate sort of approach. And so I think that there is a way of doing that, and I think it's important that those AI engineers kind of understand the problems we're coming from. Other comments from the audience, people have been working in that sort of space? Arthur?
Thank you. Actually, it's more a question than a comment. The question would be, if I want to cross-check information between the different databases, what kind of ID should I be using? For instance, if I go to PubChem, I will have the CID. But first, usually, you cannot use the name. Usually, you need to use the smiles, and even with the smile, you have different kind of smiles. So maybe some harmonization could start actually with having the same identifier and be sure that it's a unique identifier, because for now we have a lot of troubles with this. So that's the first question. My second question is, we are all using this, 350,000 figures from the Wang et al. Inventory. Could you comment a little bit about the overlap we can have between, PubChem Lite, for instance, or even the 100 million? And are we close to this inventory or stuff like this?
I'm going to answer the second question first, because I actually meant to mention that in my slides and I didn't. So this was a bit of my talk I had to leave out, because this 300,000, 350,000 that's been mentioned a few times today, that data set was never actually made available. So the paper is getting a lot of citations. The data was never released as part of it. In 0PM, we were actually working with Tanyong Wang to do a new version of the global chemical inventory. We're actually working on a website for it now from a slightly different point of view. We haven't got the overlap, but I could get it for you. It actually distilled down to about 98,000 chemicals, so it's a lot different from the 350,000. So there was about 40% UVCBs in there for which we can't even map to chemicals. And then once there's a standardized pipeline, hopefully that role will publish at some point. That is a bit out of our hands. What we're trying to do now is get at least the Norman SLE into PubChem Lite, and then we would actually look at the global inventory. The problem we've had, and the reason I introduced it with the annotation content, is there's a big section of the REACH legislation that neither the guys at PubChem can validate, Tony at EPA can't validate, we can't validate. We have a single data source for a lot of these chemicals, and these are typed in by someone, and you look at those names, and even if you have a chemistry degree, you have no idea what that chemical is. And so a lot of the other resources are actually rejecting these chemicals because there's no way to verify them, but they're very interesting because if they're on the REACH legislation, someone's producing them in at least a ton, and then we're interested in them, at least in Europe we are. So that's the one side. On your first question, actually getting back to the comment you said, so what we're seeing is, and I don't know if you're observing the same thing, when we're seeing people that kind of know a little bit about curation, but not enough about curation, and if they start providing data in standardized formats, we start to see a lot of mistakes come in. And this could actually be what those AI experts are meaning, that actually if you're getting people try to do the right thing but failing slightly, it actually introduces more problems than it solves. Whereas if they could actually just put their knowledge on a page in the way they're comfortable with, I would hope AI would soon be able to deal with that and actually take out that knowledge, make the right assumptions, and figure out the name, is this name, it means this chemical in this context, or it means this in this context, it came from that field. So I would hope AI could go there, and maybe we don't have to sit down our students and force them to do these tables and copy paste, and you would not believe how many small errors we find in our data sets, because you can tell people are copy and pasting things from different databases instead of using a resource. So what we're trying to work on now, I don't know if you're thinking of something similar, submission systems where people can provide the information they trust, and we try to fill in as much as we can to minimize those errors, or at least flag errors, so that we reduce some of these.
I think you're talking about pretty sophisticated, like scientific questions in there. Like the fact that we can get papers in the literature that have hallucinated references when there's databases to check references. Like how, like that's just a huge oversight and kind of unforced error on that part of the scientist. And I think we need to get better at doing that, because even if you're looking at some of these relationships that may be predicted by some new AI model, like things like CTD, like does it make sense? Like is there kind of this ground truth that these things are known? There is some evidence out there if they're known chemicals. And figuring out, and again, these don't seem like big leaps to say, can you double check it against this? Can you go through and make sure those things are there? And so that's many other issues.
But I think there's also a lot of resources who are now trying to, now we have to go back and feed our results to AI so that they get it right. And this seems somehow so bizarre that it can trawl through everything, and then we have to try to educate it to go to the database to find the real information. So I know there's a lot of resources struggling with this as well, so that if you would type in ChatGPT, you get the right answer, not that methanol is somehow a cyanotoxin that's ginormous. I think this has been solved by now.
I'll add a comment on the question about using identifier. So AI is pretty powerful in like fixing, connecting chemical names like synonyms and some variants of chemical names. So that problem is kind of solved. If we have chemical name, INSI key, CID, that we can all hyperlink. It's really not a big problem. The major problem is somebody says, this is the chemical X, can we validate it? And that's where we need access to the raw data. And without having access to the raw data, we can't really validate that that was the real compound. Because you can have annotation saying that this is the compound, but whoever did that annotation did a mistake, and then that mistake get propagated. So we need access to the raw data along with the annotation data, so it can be validated by an external expert that this is what it is. So for data harmonization, I think we need a national data harmonization center. for untargeted exposomics, yeah. That does everything like that, yeah.
But it would start with first, like, reporting the data.
Yeah.
Like, that would be like, the expectation is that when you're doing this sort of work, that you are depositing the right type of information into an accessible database. And again, like, I think there are some limitations to our existing systems for that we need to make better. But it is a matter of kind of get higher expectations on that.
I don't know if we still have time to jump into this is also where the regulators come in the discussions, because to get back to your identifier question, a lot of the regulation is done relative to CAS numbers, which is fine if it's in CAS and if you have a license for CAS, which is extremely expensive, so we don't work with it. But if you've got the European regulation, some of these chemicals with EC numbers don't automatically get pulled into CAS or would get into CAS with a delay, so we have chemicals that are regulated that don't have CAS numbers. We all know what Excel does to CAS numbers, so you always have a certain amount of errors in there. PubChem will accept a lot of things and reasonably quickly, but it still takes a while to get the CIDs. There's some problems with standardization, so not all of our chemicals get CIDs. Fixing that is usually manual work, me sitting at my laptop trying to fix the problem, or them trying to fix the standardization error. So there's a lot of things we can do, but there are so many edge cases. And out of that is the UVCBs, and we have still not managed to get any funding to do anything with the UVCBs to get any form of identifier that would actually work to start sharing the information on these chemicals through the resources. So we have a long way to go. I'm sorry.
Okay, so I'm going to give each of our speakers a wish. So if you could, magic wand, what would be the thing you'd want to see to advance the field of exposomics, obviously within the data sort of thing we've been talking about? What would you want? Do you want to go last because of the jet lag?
Yeah, maybe I'll go last. I got to think about that.
I'll say just thought about the National Data Harmonization Center for Untargeted Exposomics. That would be my wish.
Take notes, people.
I think the point was raised earlier about this 15-year time frame, or, for exposomics, you need something long-term. What we also need as researchers and as people do this is also the long-term infrastructure funding, so that these key resources that everyone is using actually get funding. A lot of people rely on David Wishart's results. What's going to happen when he retires? We don't know. How is that going to be funded? Things like this. Well, a lot of what we do, Stefan and I have scraped together money somehow to keep Mass Bank running. Our Japanese colleagues happen to have somehow got some money to fund their work. But a lot of this is individual researchers bring it in, and then like these discussions, how can we actually support these researchers? And probably what you'd have to do is actually switch out the researchers. So, if something fades away, how do we keep that innovation up? You have to fund what's keeping going, but ensure that there's some solidity so that the workflows can actually be standardized and run, I guess.
Can we have two wishes?
Well, let's let Carolyn go first, and then you can try another one.
Yeah, God, there's so many things I wish for, but yeah, I think the longevity of the projects is really important, because NIH, at least in our case, makes these investments, but every 3.5 years, you're not sure if you're going to exist again. And not that you stop working on it, but there's a real risk of what's going to happen to all of these investments and all of these data. And I think, all right, I'm starting to weave into another one too, but if there was I know there are efforts that have been born out of conversations at NIH of harmonizing projects, and we talk about it in the DRKB group. But I think there's still a lot of really disparate resources out there, and figuring out some real substantive way to get us more connected, because we're all linking to each other in certain ways, but there's a lot more that could happen if that integration was more substantive, I think.
Okay, what's your bonus wish there?
Yes. So we have IARC monograph for carcinogens. We really need a similar monograph program for neurotoxins.
Okay.
I've been saying that we really have to have a monograph for neurotoxins.
Okay. On that note.
Time for a small question?
Small question, but maybe not an answer. Go ahead.
Thanks. So on the topic I just recently listened to a presentation where in human genetics they put together data of more than 5 million people, and they said the data saturated, so it's like too much data, it's enough. How about exposomics, and we go on chemicals? Some of us do have samples on very long term, but then to be afford to run through all the throughput analysis, how much samples and data we need?
So I don't think we can answer that in our given time.
Does anyone want to do a
sounds
power
like
calculation
a
on
great boat sort of conversation this evening about how much data do we need. I think that is a key overarching question. So on that question, I will turn it back over to Carla, whoever's going to say how to get to the boat.